Actinomyces israelii. General information. Actinomyces israelii, taxonomy and significance. Taxonomy. Actinomyces israelii belongs to the family Actinomycosis. This family encompasses a group of bacteria that are often filamentous and exhibit branching patterns reminiscent of fungi, hence the name Actinomyces. Natural habitats. Interestingly, this bacterium is not an environmental organism, it's a natural resident of the human body, particularly within the oral cavity and the gastrointestinal tract. It exists as part of the normal flora, usually without causing any harm. Clinical significance. While generally harmless, A. israelii has the potential to turn opportunistic. It's the primary culprit behind actinomycosis, a chronic infection notorious for causing slow-growing, painful abscesses. These abscesses frequently manifest in the face and neck region, but can also affect the lungs or the gastrointestinal tract. A hallmark of actinomycosis is the presence of sulfur granules within the abscesses. These granules are actually dense aggregates of the branching A. israelii bacteria, giving them a characteristic yellowish appearance. Notably, actinomycosis often involves other bacteria in addition to A. israelii, creating a polymicrobial infection. Actinomycosis, the disease. Actinomycosis progresses insidiously, with abscesses enlarging over weeks or even months. This slow growth can lead to delays in diagnosis and treatment. The abscesses associated with actinomycosis are not only painful, but also tend to form fistulas or tracts that burrow through tissues, potentially leading to disfigurement or complications depending on the location of the infection. The presence of sulfur granules in the abscesses is a key diagnostic clue for actinomycosis, though laboratory culture and identification are necessary for confirmation. Gram stain. Actinomyces israelii is a gram-positive bacterium, recognized by its branching rod-shaped structure. This branching morphology often results in the formation of dense clusters known as sulfur granules in infections. Culture characteristics. Actinomyces israelii thrives in environments with low or no oxygen. While it may initially struggle to grow in the presence of oxygen, it can adapt after several subcultures. On blood agar, its colonies can be rough, resembling a molar tooth, or smooth. These colonies do not cause hemolysis, breakdown of red blood cells. On Brucella blood agar, rough colonies develop the distinctive molar tooth shape, while smooth colonies appear creamy and grow faster. About 1/3 of strains exhibit these smooth, fast-growing colonies. Regardless of the initial colony type, older Actinomyces israelii colonies may develop a pinkish hue. Characteristics. Actinomyces israelii is a gram-positive bacterium, displaying a characteristic branching filamentous morphology under the microscope. This bacterium exhibits flexibility in its growth, capable of thriving in both aerobic and anaerobic conditions. Biochemically, it tests negative for catalase and oxidase enzymes. Further distinguishing features include its non-motile nature, susceptibility to vancomycin, resistance to colistin, and the formation of sulfur granules in infections. When cultured, its colonies present a unique appearance, described as either molar tooth or smooth. Poem. In shadows deep, where oxygen's scarce, Actinomyces, a branching farce. Gram-positive, its filaments entwine, sulfur granules, a telltale sign. Molar tooth or smooth, its colonies dwell, a microbe's tale, its secrets tell. From harmless guest to abscess ire, Actinomyces, a hidden fire.